And our final student speaker will be Boxiao Li, who's a PhD candidate in the Energy Resources Engineering Department at Stanford. He's focused on bridging the gap between small-scale heterogeneities and reservoir-scale simulation in order to better predict long-term fate of sequestered CO2. Also awarded the Frank G. Miller Fellowship, and he's been one of the student representatives at Stanford to help build this program and others associated with GSEP. So welcome, Boxiao.
Okay, thank you, Sally, for your introduction, and thank you for GSEP for giving me this opportunity to present some of my research to all of you. So, I don't think I need to motivate why CO2 sequestration is important. It is one of the most effective ways to reduce anthropogenic CO2 emissions into the atmosphere. So after CO2 is captured from sources like power plants, it is transported to and injected into the underground geological formations. So in this research, we focus on sequestration in saline aquifers because they are widely distributed around the world and they have a big sequestration capacity. So in order to accurately predict CO2 migration in the saline aquifer, we need accurate simulations. So the flow of CO2 and water occur in the tiny pore spaces in the formation rock. Now, however, it is intractable to model a large aquifer by tracing fluid migration in the tiny pore spaces because there are numerous pores in an aquifer. So Darcy's law, based on continuum assumption, was developed so that people can use that to model flow in porous media at a larger scale, like the core scale. Now, the core scale is very important because we can collect these cores from drilling and then perform high-resolution, accurate, and affordable measurement at core scale to reveal the physics. And however, it is still computationally unachievable to model a large aquifer based on core scale because you may need trillions of grid blocks to do that. So when we are modeling a large aquifer, the typical number of simulation grid blocks is around 1 million, such that it is computationally achievable, and each simulation grid block is on the order of 100 meters. But the challenge is that we cannot perform high-resolution, accurate, and affordable measurement on such a large scale. So in order to accurately simulate a large-scale aquifer, we need to start and do rigorous study on a smaller scale which we can measure, and namely core scale, and then translate this important physics into a larger scale. However, in the common industry practice in translating from core to aquifer scale, people usually ignore the small-scale heterogeneities inside the core. And this means that there are some important physics may be lost during this translation. So our goal is to bridge the gap and ensure and preserve the important physics during the scale translation. So we ask ourselves several questions. Now, how do we model fluid flow in core scale? Basically, it means what's the physics in core scale flow? And how important is this small-scale heterogeneity in a large-scale simulation? And if it is important, how can we include it and translate this important physics in large-scale simulation? So our approaches are, we first perform core flood experiments to reveal the fluid flow and the physics in core scale. And then we perform high-resolution simulations trying to reproduce the experimental results and then understand the core scale physics. And then we can use what we understand to study the impact on larger scale. So the core flood visualization lab we have at Stanford University employs the X-ray CT imaging to accurately measure fluid distribution inside the core. And this is a typical experimental result. What I'm showing you here is the CO2 saturation distribution map inside the core measured by CT scanner. The definition of CO2 saturation is the volume fraction of the CO2 in the pore spaces. As you can see, the red color indicates the region where CO2 saturation is high, and the blue color indicates the region where CO2 saturation is low. And the CO2 saturation is distributing in a very heterogeneous manner. And we have experimental evidence as well as simulation effort to say that this is caused by spatial variation in the capillary pressure saturation relationship, also called capillary capillary heterogeneity. So this means that if we want to accurately model this core flood process, we need to account for capillary heterogeneity in our simulation. And however, performing these core flood simulations are very difficult because first, it has high resolution. Now, this plot is a typical simulation grid we use in our simulation. In this picture, we have over 50,000 grid blocks in there, so it's a very large model. And also, we need to account for capillary heterogeneity. This means that each simulation grid block has its own capillary pressure curve. So in this plot, we have over 50,000 capillary pressure curves, so it's very complicated. And also, the simulation time is very long. And we were able to perform this high-resolution simulation using our previous simulator, TUF2, but each simulation took us roughly 4 days to finish in a computer cluster, so it's very slow. And also, we were not sure whether we were using the most effective numerical strategy. And neither were we sure that the numerical approaches we used gave us the correct answer. So before we can study the scale translation problem and to account for the small-scale heterogeneity, we first need to have a rigorous mathematical and efficient numerical framework that models capillary heterogeneity correctly. So from a numerical standpoint, to model a physical process correctly, we need to honor 3 key elements. Mass conservation is honored for each simulation grid block as well as for the entire simulation domain. And the flux is continuous across each simulation grid block interfaces. And the flow direction at each grid block interface is correct. But the challenge of capillary heterogeneity is such that, first, the strong nonlinearity introduced by the capillary pressure term it makes it very difficult to solve the mass conservation equations efficiently. And second, the discontinuity and the heterogeneity of the capillary pressure functions poses significant challenge in honoring flux continuity. And also, the complicated interaction of viscous buoyancy and capillary pressure forces troubles the simulator to find out the correct flow direction. So we have identified and employed the correct numerical approaches that to tackle these 3 challenges at the same time, such that these 3 key elements are satisfied. And with that, we implemented the functionality of capillary heterogeneity inside our in-house simulator, the Stanford General Purpose Research Simulator, or GPRS. Each simulator has to be verified with a benchmark. Now, fortunately, we have a simple benchmark problem of capillary heterogeneity that has a semi-analytical solution. So the model is composed of 2 regions with different capillary pressure curve. The initial saturation distribution for the 2 regions are very different, and they are shown in this picture. And now, starting from this initial saturation, the semi-analytical solution depicts the change of saturation distribution over time. And as we can see, due to capillary heterogeneity, there is a saturation jump across the region interface, and we need to capture that in our simulation. So our simulator, GPRS, produces the simulation results shown by the circles, and they match the semi-analytical solution very well. And particularly, it is able to capture the saturation jump across the interface. So this means that our simulator is doing the right thing so that we can use that to model our core flood process. So here I show you some of the results. What I'm showing you here is the CO2 saturation distribution map taken from an arbitrary cross-section across the core. And I am comparing the simulation results versus the CT scanning results measured in the experiment. Now, please pay attention to the pattern of how CO2 is distributed inside the core, and also pay attention to the value of CO2 saturation depicted by the colors. If you can compare these 2 plots, and you can see they actually match each other pretty well. And if you plot these 2 data sets together, the correlation is indeed very good. So this is telling us our simulation has very good accuracy. It is able to capture the pattern as well as the value of CO2 saturation distribution that is consistent with the physics. And furthermore, the simulation time is greatly reduced. And previously, it took us 4 days in a computer cluster. Now we spend almost 1 hour to finish in a personal computer. So it's so it means that we now have a very efficient and robust numerical tool for us to do a lot of things. Now, our next research question becomes that how important is this capillary heterogeneity in large-scale simulation? Now, if we look at the permeability distribution inside this particular rock, the first thing we may notice that there is a low permeability lamina running through the core. And our intuition tells us that this low permeability lamina may be a flow barrier of CO2 that's going to affect fluid migration significantly. And this is indeed the case. We have experimental results, as well as this type of behavior is reported in the literature. However, in the remaining region of the core, although we do see heterogeneities in there, the heterogeneity appears to be relatively homogeneous, and it does not form any distinctive structure. Our intuition tells us initially that this relatively homogeneous heterogeneity may have very limited impact on fluid migration. However, this is not necessarily the case. If we look into this 2D vertical domain, 1 meter by 1 meter, and each simulation grid block is 4 by 4 millimeters square. And apparently, this domain is relatively homogeneous, but if you look into the detail, you do see a heterogeneous distribution of permeability. And also, each simulation grid block has its own capillary pressure curve, so it's also capillary heterogeneous. If we slowly inject CO2 from left to right to displace water, and the injection rate is slow such that it mimics the CO2 flow rate in the post-injection period. This is the simulation result of when we are considering capillary heterogeneity. As you can see, due to gravity, the CO2 tend to move upward and form a gravity tongue. However, the advancement of the CO2 front is not that easy. CO2 encounters some local obstacles, and that's why the displacement front is jagged. This means that the CO2 mobility is restricted by capillary heterogeneity. Now, if we keep all these simulation parameters unchanged, but ignore capillary heterogeneity by implementing a homogeneous capillary pressure function inside our simulation, evaluated as the average of the domain, this is the simulation result we get. And in this case, the CO2 mobility is much greater and is happy to move forward and upward and form a bigger gravity tongue. And the plume extent of the CO2 plume is almost twice as large as the case when we are considering capillary heterogeneity. So this example tells us that capillary heterogeneity is indeed very important, and we need to account for that. But the practical problem is that in large-scale simulations, we cannot afford to implement so many capillary pressure curves inside our simulation. So we have to come up with some effective multi-phase flow properties to use in our simulation. How do we calculate these effective properties while accounting for the impact of small-scale heterogeneity is our ongoing work and will continue to be our future research direction. So conclusions. The small-scale heterogeneity can be very important in large-scale CO2 migration. And by performing core-scale and sub-core-scale measurement and simulation, we are able to gather very valuable information about core-scale physics, and that can be used in translating into a larger-scale simulation. Now, we have this efficient and robust numerical tool that allows us to characterize small-scale heterogeneity, and also serve as a foundation of our understanding how do we translate this small-scale information into large-scale simulation. We are working on that right now, and it's going to be in our future research direction. So finally, I would like to thank GSEP for financial support, and I would like to thank the entire team for their help. With that, I'd be happy to take questions. Thank you. Yeah, I know you're going to ask questions.
We would be disappointed.
Well, you gave a very nice talk. Thank you.
Yeah, thank you.
Couple of questions. Actually, I have more. I'll talk to you one-on-one.
All right, sure.
The picture that you showed where you actually match experiments with populations, obviously you adjusted something like a permeability or an entry capillary pressure at every grid block. In other words, you had a number of adjustable parameters, essentially at least one per cell, to match the experiment with the theory, probably 2 per cell, right?
Well, I mean, which plot are you talking about? The experiment?
You had the experiment matched with the cross-sectional calculation. You had 2 circular cross-sections.
That one?
Yes.
Okay.
So obviously you had 2 parameters at least adjusted in each grid block. Which that match, right?
Well, in terms of the simulation parameters, we calculate basically the scaling of the capillary function is the J function scaling, and we measure that from the experiment. And then the porosity we measure from CT scanning, and the permeability we measure from also CT scanning using the approach developed in our group, Dr. Mike Krause. So that's the simulation parameter we use in our simulation.
But it's basically inferred through some indirect means for every grid block: permeability, entry, capillary pressure, and porosity. I presume. Okay. So the other question is actually a more deep-rooted physics question, which is that when you look at the length scale on which you're introducing these capillary pressure functions, you've got, in a 4 millimeter by 4 millimeter cell, you've got subgrids and so on. Yeah. And then I start to ask, do you actually even have a mean field approximation when capillary pressure varies and that kind of a length scale, because you have something like hundreds of pores, and to define a capillary pressure you need literally 10,000 to 100,000 pores in order to have the correct mean field approximation. Yeah. So you are at a crossroads where you can't even define continuum properties anymore. Oh yeah. Oh, that's a very good question. I'm very glad you mentioned that. And a lot of researchers have done this. What's the size of the representative elemental volume? I don't have the picture right now, but there are a group of scholars have done that, at least for Berea sandstone and other similar sandstone. Like 2 millimeters or 1 millimeter is enough to be account for a representative elemental volume. It's reported in the literature, and I can show you if you're interested. I certainly agree with you on single-phase flow, but I'm not sure about two-phase flow.
Multi-phase flow. I'm not quite sure that in that regard, or we can discuss later. Yeah, thank you.
Okay, one more question, and then we need to move on. Yeah. Any questions? No? Okay. All right. Well,
thank you.
Thank you very much.